It's my pleasure to introduce Dr. Andrea Small-Howard. She has more than 20 years of scientific research and executive experience in the biopharma industry, supervising research and development, manufacturing, and quality control divisions in the U.S. and China. Dr. Small-Howard has taken novel biological products from ideation through commercialization. She has been named an inventor on more than 60 patent applications, taking the lead in obtaining regulatory approvals in the U.S. Food and Drug Administration and numerous international regulatory agencies and orchestrated licensing deals. Currently, Dr. Small-Howard leverages her broad biopharmaceutical industry knowledge as the Chief Science Officer and member of the Board of Directors of both GB Sciences, Inc. and their wholly owned Canadian subsidiary, GBS Global Biopharma, Inc. Dr. Small-Howard brings her passion for advancing research on phytochemical compounds to her current roles. She envisioned bringing more phytomedicines to market through an innovative AI-based drug discovery engine and biopharmaceutical drug development program for disease-specific indications. So without further ado, welcome and look forward to your talk. Thank you.
Thank you very much. Can you see my screen right now? Excellent. Because I can only see my screen, but that's okay. That's what we need, huh? So I'd like to thank the team at Precision Global for inviting me to talk today. This is a topic that I'm very passionate about, and I hope to sort of spread the word that plants are an excellent source of novel drug compounds, and which is not something that's new, but it's something we're revisiting. Anyway, so as we mentioned, my name is Andrea Small-Howard. I'm the Chief Science Officer for GB Sciences. GB Sciences is a phytomedicine company that is developing combination drugs for the prescription drug market. Our discovery program is able to create unique APIs and a growing intellectual property portfolio. Our discovery program combines the use of both GB Sciences' proprietary AI-enabled drug discovery platform, as well as some good old-fashioned natural products research. When we take things from discovery into development, that's where we start to use more traditional sort of biopharmaceutical type strategies. So instead of using plant-based compounds that we use in discovery, when we go into development, we're using synthetic homologs. So these are identical to the plant compounds that we originally were characterizing, but they're now being produced synthetically under CGMP regulatory environment. We believe that'll be a helpful strategy for those regulatory agents that are eventually going to see these formulations. In addition, we are combining these synthetic homologs of the plant compounds with proprietary oral delivery formulations. A lot of the plant compounds that are in our APIs are very lipophilic, and therefore delivery is something that we need to address early on in the process. So we have selected oral dissolving tablets, oral thin films, oral nanoparticles, and gel capsules as for our different programs. In our drug development pipeline, which I'm not really going to talk too much about today, our lead program is in Parkinson's disease. The patent has been issued in 2020, and we believe we'll be IND ready by Q2 of 2022. And our next program is in neuropathic pain. The patent was also issued in 2020, and our neuropathic pain formulations are in preclinical animal trials at the NRC in Canada right now. We have two anti-inflammatory programs, actually. One is for a rare disease called MCAS, and the patent was also issued in 2020, and it's animal proof of concept ready, as well as we have a program that is creating drug candidates to address the inflammatory properties of cytokine release syndrome. The patent's been filed, and it's in a proof of concept study at Michigan State right now. This is just a graphic display of the information about our drug development pipeline that I just mentioned before. As I mentioned before, our Parkinson's disease program is taking the lead. We're really trying to get to our first in man as soon as possible. We've got neuropathic pain and our heart program that are both deeply in the animal phase. We're doing work also in preclinical on our cytokine release syndrome therapeutic, as well as our mast cell activation syndrome. And we have an inflammatory bowel disease formulation as well. So today's topic is really about the rational design of plant-inspired mixtures. So when I say plant-inspired, what do I mean? Well, as I mentioned before, during the discovery part of our drug pipeline, we're really looking at natural products. But when we convert to a development program, that's when we start to use synthetics. So that's why I always consider these plant-inspired mixtures rather than plant-containing mixtures. So what do I mean by rational design of plant-inspired mixtures? Well, when we're talking about plant extracts, they have, depending upon the method you're using to measure them, they can have literally 1,000 compounds in there. So when we're talking about formulation, you do not want that kind of complexity. So the challenge that we encounter is if you've got a plant extract with therapeutic potential, how do you come up with a minimum essential mixture that you can reproducibly produce and validate through the US FDA process? And so the other thing when we're in this process that we don't want to do is we don't want to throw the baby out with the bathwater, right? So although we are simplifying our disease-targeted formulas from this plant extracts, we want to make sure that we're preserving the molecular synergies from the plant that increase the effectiveness of these individual ingredients while reducing the complexity to the minimum essential. Part of the keys for being able to make sure that we are working with the minimum essential mixture is that we are spending time to identify the mechanisms of action and to study the therapeutic interactions between multiple receptor-based reactions, which is the disease states that we're really targeting. It's not single receptors. So unlike a traditional pharma program where you'd be looking at a single receptor target and looking to find a single high affinity ligand, we're really talking about disease processes where there's multiple receptors in play, and we're looking for a combination of lower affinity ligands that will play well together and will help us accomplish our clinical outcomes. And in this process, we're also looking from the onset to try to predict competitive and side reactions. And based on the program that we have, we believe that we can accomplish all of these things. So the big picture of our drug discovery platform, as I mentioned before, is two components, and they're both really important. One is our in silico convergence analysis, and which is an AI machine learning enabled drug discovery platform, which uses both data analytics and machine learning and deep learning algorithms in combination with data that we're generating through high throughput screening systems, including those that are disease-specific cell and animal models, as well as going to outside sources to supplement that data. The outcome that we're looking for are these plant-inspired optimized therapeutic mixtures, and we then patent these. A little bit about the drug discovery platform. I'm not going to spend too much time on this because I was on a panel yesterday, but basically the idea here is, so in the early days, we were looking within the cannabis plant and trying to deal with the complexity in that plant. We've now sort of opened it up, and we're now looking at all plants as sort of the playground for our drug discovery. And so we've set up a tool that we call Ferrous, which stands for Phytomedical Analytics for Research Optimization at Scale. It's both a science gateway and a virtual research environment for drug discovery. It includes transformative data integration, analytical methods, and visualization tools for the meta-analysis of non-Western medical knowledge systems, also known as sort of traditional medical pharmacopoeia. One of the key units is the idea that we use convergence, because sort of the big idea is that if over thousands of years, people had been using traditional medical systems to help treat their symptoms, and they were getting some efficacy there. If you have these happening in separate places all over the world, those areas where there's convergence or where they overlap, there should be some increased probability that you'll get efficacy. So in other words, the places where they share compounds or share families of compounds, there should be a greater probability that would be a useful therapeutic. And so the program is set up and it pre-validates the efficacy of drug target indication relationships, and it's been trained to predict novel compound drug candidates from within traditional medical systems. But we also do a lot of just traditional screening like you would for any other drug discovery program. So we've done a lot of high throughput cell models for the different indications. Some of them are here on the slide. In addition, I've also put down some of the assays that we've used for to come up with to do different levels of screening of the different formulations. And another thing that's been incredibly useful for us is that we've been using a lot of a high throughput animal model, which is the zebrafish. And it's not good for every indication, but for a lot of indications, you can do a lot of sort of intermediate work as far as eliminating formulations that don't work by putting them in these animal systems. And you get the complexity of an animal, but you get that rapid turnover and high speed and rich data sets by using the zebrafish models. In our intellectual property, as I mentioned before, we're really looking to identify plant-inspired optimized therapeutic mixtures. And these are a novel API composed of natural or synthetic homologs of plant-derived ingredients. We're claiming both composition of matter and field of use claims. And our newer patents are claiming not only the novel API, but also combinations of these novel plant-based API mixtures and novel delivery methods. Our current portfolio contains 5 issued U.S. patents and 3 issued international patents, as well as 7 non-provisional and 35 7 non-provisional U.S. and 35 non-provisional international patent applications and 3 U.S. provisional patent applications. And as I mentioned before, with these plant-based compounds, we wanted to get ahead of the idea that because they're very lipophilic, it's great to have drug delivery systems that are going to increase the bioavailability and increase their absorptivity. And so we have been lucky enough to partner with other folks that have proprietary delivery methods, including we're using the Zydis rapid dissolve tablets from Catalent and their oral gel cap formulations, as well as some novel cannabinoid nanoparticles from the University of Sevilla and the University of Cadiz. And so I thought at this point, I'd give you sort of an example I'm giving you 2 examples. One is for how we generated our Parkinson's therapeutic candidates, and the other one will be how we generated our novel neuropathic pain candidates. So this is another illustration of sort of the funnel that we go through, in that we started off by doing a lot of work with the cannabis plant. We were looking at over 2,000 metabolomic profiles. And then we were using some modules and some random learning modules combined with some cell data just to eliminate the potential mixtures. Because if you looked at all of the compounds and all of the different metabolomic profiles for cannabis that we were looking with in our original data set, there were well over 10,000 potential mixtures there that looked promising. We then reduced that using our drug discovery platform to around 1,000 combinations that we went ahead and did the high throughput cell screens, where we were looking at dopamine release and protection from MTPT, which is a neurotoxin that mimics the state that you're in a lot of neurodegenerative diseases, including Parkinson's, Alzheimer's, Huntington's, and Lewy body dementia. And from there, we took the most promising mixtures. And we had to, so at the level of the cell data, the combinations that we were using had about a dozen active ingredients in each of those combinations. In going from the cell to the animal, we were able to reduce it from a dozen active ingredients down to between 3 and 5. And we believe that in the 3 to 5 range, you get the full benefit of having synergy among the molecules without the complexity involved in the manufacturing, the QC, and the regulatory approval that will someday be on our horizon. And then, so the middle stage was actually our receptor physiology. We did some docking studies, and we did some inflammatory profiling. And then we went into the zebrafish. And so from the zebrafish, we ended up with, at the end of the zebrafish data set, we ended up with just 3 mixtures with specific ratios that are now sort of the lead. And then we took those they're now in rodent testing. So to take you back to sort of some of the highlights of the data that we learned along the way, this is one of the data sets from our early studies where we were in the cell models. And early on, we asked ourselves, is the power in these plants an individual ingredient, or is it a mixture? You hear a lot about how it should be mixtures, but there weren't a lot of data to support that. So we went ahead and set up these experiments where we basically were taking the ingredients from the plant and setting them up as these mixtures that we could test in a very discrete way, so that we could test all of the individual ingredients, and then we could test them in mixtures that we put together ourselves. So one of the things that was very obvious to us in looking at this data is that while there certainly were some significant signals in the individual ingredients, they were dwarfed by the mixtures. The part that was surprising to us in the early days is that the prevailing literature led you to believe that you would need a quote unquote major cannabinoid, or one of the ones that was at was in high abundance in the plant. So THC, CBD, or CBN, to have full effectiveness. But what was really surprising to us in these early days is that the complex mixtures that contain no quote unquote major cannabinoids, lots of minor cannabinoids and terpenes and other ingredients, still had a lot of effectiveness, which was then amplified by adding a major cannabinoid. And in this particular scenario, we tested the THC as a major cannabinoid, and you didn't get any difference relative to the complex mixtures without a major cannabinoid at all, which we thought was interesting. So then when we were going into, as I mentioned before, we took the lead combinations into a zebrafish model of Parkinson's using OHDA. OHDA is a very well-established neurotoxin used to mimic the dopamine activity, or reduction of our dopamine activity in animals. And so it's used as a Parkinson's model quite frequently. And what we were able to do in the zebrafish model was we restored the overall movement levels relative to the normal untreated. And we were able to reduce something like the resting tremor, as well as we retain a normal startle response. So the exposure to our compounds were not having an effect that was overall sedating or anything. It was truly helping in a much more targeted way. And so this is a small sample of a large data set that we're going to be publishing later this year, which demonstrated the statistically significant reduction of these Parkinson's motor symptoms. And so the parameters that you're looking at here, so you've got your control bar in panel A, and then you'll see next to it is the drug and the OHDA. So that's the experimental system. You have your drug alone, and then you have the OHDA alone, which gives the most dramatic symptoms. And so the first one is looking at, so you use actigraphy to actually show the twitch. There's actually a demonstrable twitch that you can see. And so A is looking at the frequency of switching from twitching to non-twitching. The next is the amount of time it spends in the on state versus off. And the last one is looking at just overall, how well are these animals moving? And so we see an improvement in all of these parameters with one of the three drug formulations, OTM PD119. One thing that's significant too is that we're able to achieve these reductions in Parkinson's-like symptoms using a submicromolar level. The total molar concentration of the mixture is half a micromolar. So the individual components are less than that. And then, as I mentioned before, in development, we're going to be taking these forward in the orally disintegrating tablets that are our Catalent's Zydis proprietary formulations. What we like about why we think that these ODT are a great match for this therapeutic is that they're a unique freeze-dried dosage that you just put on your tongue and they dissolve within 30 seconds. It's super convenient for Parkinson's patients because greater than 50% of Parkinson's patients have problems with swallowing. And clinically, it should have advantages. It's improved bioavailability as well as compliance. If your patients won't take the medicine, then you're not going to see any results. And it has rapid onset through the buccal sublingual absorption. So that's the story for our Parkinson's. So then how do we use rational design principles to come up with our neuropathic pain formulations? So I'll take you through that scenario real quick. So high level, I wanted to put this proof of concept slide up first. So this is taken from a study that we've already published. And what it shows is that if you take unique active ingredients and you put them in one of these extended relief oral nanoparticles, you can get a significant potentially significant clinical benefit. So this study was done using a rodent model of neuropathic pain. And in that experiment, what they did was they compared the effectiveness of taking the free cannabinoids versus cannabinoids that were inside a nanoparticle with this oral time release property. And so what you saw was that a single oral dose of the cannabinoid-containing nanoparticles was able to provide sustained relief for 11 days, whereas when they applied under the same conditions, the free cannabinoids, so not encapsulated, it had less than a day. It was really a peak activity at about 8 hours. And so this illustrates not only the potential, but sort of where we're taking our pain programs. We're combining the novel active ingredients with the extended relief strategy. So at its core, the strategy behind our neuropathic pain formulations is summarized in this slide. Basically, within a nociceptive sensory neuronal bundle, the different neurons express multiple channels, TRP channels. So this is a family of channels, transient receptor potential channels, and they respond to different things like hot and cold, but also chemical sensations and all kinds of things. And so these have been linked to pain responses. Conventional capsaicin-based pain therapies only target one of these TRP channels and leave the other receptors in the neuronal bundle untouched. So our strategy, and what has been supported by both our in silico network pharmacology as well as our cell-based experiments, revealed that our mixtures have the potential to target multiple of these TRP receptors in the bundle to increase their net effectiveness. And so with this one, we really took that natural products approach, like I mentioned in the beginning. We started with, in this scenario, a single variety, or what is known as a single strain of cannabis in the sort of the vernacular, where patients were saying that it was very helpful in relieving their pain. And so based on the anecdotal reports from our patients, we decided to use this one as a potential drug candidate. So we went through and panel A, so we were using a cell-based model to assess whether these mixtures could trigger a TRPV1 response. So I'm showing you the TRPV1 data, but we did this with all of the TRP channels. And so panel A this is the nature of a positive response done with capsaicin, just to show you sort of the magnitude. And then on panel B, what you see is the result if you take the whole plant extract and you put it into this cell-based system, you get a very strong and robust TRPV1 signal. As a first approximation to establishing where the activity was, we were able to easily separate the terpene molecules from the cannabinoid molecules, and we tested each of those fractions separately. We found that the cannabinoid fraction had a significant response at TRPV1, but it was dwarfed compared to what we saw when we looked at the terpene fraction. And so we decided to go down and use sort of a reductionist approach and look for, in this scenario, we were testing all of the terpenes in that strain individually at the levels that they were at versus if you added all the terpenes together. And so you see that if you add all the terpenes together, the line in blue does give you the strongest response. But a large proportion of this comes from, of that signal comes from myrcene, and some from neurolidol, and some from the others that although they are small when done separately, when used together, there is synergy. And so we took the most effective mixtures of these terpenes and we put them into surface-modified PLGA nanoparticles with a time-released property of these chains on it, which are the same. So basically, we set up the same scenarios I showed you in the first slide. We put the terpenes into the time-released oral nanoparticles. And one of the things that we did as a strategy is we put each of the terpenes in separately so that we could change up the ratios in an experimental system. And so in the early days, we were just testing to see, okay, how well do these terpene encapsulated nanoparticles perform relative to the free ones, which are not encapsulated in the same TRPV1 cell system as I showed you before. And across the board, the nanoparticles had a stronger immediate effect, and when we take it out, they also have a stronger sustained effect. But then we were also interested, of course, in how to establish whether there were synergies. And so the interesting thing here is we were able to see both positive and negative synergies in this kind of data. So as well as it also aligned with the data that we got out of our molecular docking studies at the receptors. So one of the things that we knew going into this is that both myrcene and neurolidol bind to the TRPV1 receptor at the same activation site, whereas beta caryophyllene binds at a different site. And so, not so surprisingly, so panel A has the traces for both. They chose the reactions from myrcene alone, neurolidol alone, or the combination. And really, the combination is just the same as what neurolidol looks like because they both are competing for the same site. In contrast, if you look at panel B, you have the myrcene alone signal, which is in green. You have the beta caryophyllene signal, which is in blue, and the combination does show positive synergy. In contrast, when you go down to panel C, the neurolidol signal is in yellow, the beta caryophyllene signal is in blue, and the combination is in green. So you actually see negative synergy there. And then when you start to add them, you do get interesting patterns. And so when we were rationally designing these mixtures, we were obviously trying to maximize the positive synergies, minimize the negative synergies in our final formulations. And so we've now taken our final formulations and they're being tested in the zebrafish model of nociception. We're testing single compounds, both within the nanoparticles and not within the nanoparticles, and that work is done. We're now testing the mixtures, both within the nanoparticles and not in the nanoparticles, and it's looking very promising as far as being able to be able to preserve that synergy that we saw in the cell system. All of this work would not be possible without the excellent collaborators that we have established across the world. We work very closely with Chaminade University, Michigan State, and the University of Athens on the discovery side. We work with the University of Sevilla and the University of Cadiz on the nanoparticle work. We work with the NRC in Canada and the University of Lethbridge and the University of Hawaii all on our animal testing. We're very fortunate to work with Purcis that creates that creates our synthetic homologs of the plant-based compounds for us under CGMP regulatory environment, and Catalent for their help in providing our CMC and they're making our clinical prototypes and helping us get ready to bring those to the clinic, our Parkinson's formulations to the clinic. So in summary, by using rational design principles, we are able to create multi-component drugs based on plants using our proprietary drug discovery engine which creates these optimized therapeutic mixtures. We also combine that with high throughput cell and animal models of disease. Our active ingredients start off as plant ingredients, but we are using synthetic copies of these plant compounds in our formulations as they go through development. Our lead programs are advancing to the clinic, and we're expanding the potential ligands that we're using in our discovery to include the traditional medicine pharmacopoeia. And that's all I have to say today. Are there any questions?
All right. Yeah, thanks so much. Wait for the questions to come in.
So we'll open up the floor for any questions for Dr. Andrea. Her group is doing a lot of amazing things, revolutionary stuff that they're definitely doing in their drug development program. So we'll just give the floor another minute or so for any questions to pop in. And Dr. Finnett, if you have any remarks, feel free.
Yeah, sure. I guess I was most interested in the zebrafish studies. It's a really, if you could kind of apply that to lots of our other areas of drug discovery, I think that would be it's a lot easier than waiting for, mouse studies to complete, or go through all that expensive,
Yeah, I'm a true believer in that. I got to tell you, the amount of data, there's just the sheer amount of data we were able to generate so quickly. It would have taken us several lifetimes to do that. You know what I mean? It's just,
no, absolutely. And you think about it, the mouse, mouse models are not, it's not a human system anyway. Even if they humanize it, it's not, how close is that mouse to human rather than the fish? So it's the zebrafish. So to me, it's probably screen tons and tons of compounds in a real, full live organism rather than, just starting off with some cell culture. So that was very, very, very interesting.
And it's like I said, so I'm a big fan of using that zebrafish as sort of an intermediate step. We've bought into, we're taking our lead compounds and putting them through whatever the mouse model of the day is so that the FDA is happy. But I'm a firm believer that the zebrafish data is just as valid. You know what I mean? So
yeah, it's the same mechanism and it causes the same type of effect. What's the difference? So. But
it's nice because, like I said, the zebrafish really helped us. We were able to do so many different runs, and the data is so rich. Each data point is the combination of 25 individuals. And so it is an incredibly rich data set. So you can get those little subtleties. It's like doing a phase 3 trial, right? You've got so many, so robust, and the data set is so robust. And then what we were doing is then, like I said, the lead candidates that came out of the zebrafish we're putting into a mouse model to sort of hopefully just validate what we saw in the zebrafish. So that's kind of where we're at. So. But I encourage people to explore those zebrafish models. I think they're going to be incredibly useful for other programs as well.
Yeah, absolutely. Thanks.
Oh, wonderful.
All right.
All right.
Thank you so much, Dr. Andrea.
Thank you.
Yes, thank you. Yeah.
And Dr. Finith, I guess we'll all head over to our second last presentation with Dr. Tiffany Bowe.
Sounds great.